Okay, and my slides are already slightly worryingly, the text is dropping off the bottom, so we'll see how we go. So yes, this is a more biology focused with bits of Nextflow here and there. So hopefully you'll get both the biology story, but also the Nextflow bit. Hoping I've balanced it right. So yes, I'm going to talk about circulating cell-free DNA methylation and using that to detect and subtype cancer. I'm part of the Cancer Research UK Cancer Biomarker Centre, which is connected to the Cancer Research UK Manchester Institute and is all part of the University of Manchester. Okay, so circulating free DNA is bits of DNA that's just floating around in the plasma in all of you. So everybody has some, but this is increased in patients who have cancer. And some of that increase comes from the cancer cells. And so you can study the cfDNA, the circulating cell-free DNA, to learn something about the cancer. And these methylation changes often occur early in the cancer development, and the amount of tumor fraction in a patient, the amount of cfDNA that comes from the tumor can be very variable. And one of the things about this methylation changes is they typically occur early on in cancer development, and they can be quite consistent between multiple different patients with the same cancer type can have the same changes, whereas mutations, they might have very different driving mutations. So within the Cancer Biomarker Centre, we developed an optimized methyl capture sequencing approach to profile the whole genome-wide cfDNA methylation patterns. So we take the DNA, we add on at the very first step. So this cfDNA that you can extract from plasma can be as low as 1 nanogram in a blood tube. So it's very low amounts of DNA. So what we have to do is we add on a sample barcode so we can pull the samples as the very first step. And then we can perform, like we have sufficient material to perform a methylation enrichment using a methyl binding domain protein in order to pull down the regions of the genome, the fragments of the DNA that are methylated. And then we can profile that. And this is a bit like ATAC-seq, ChIP-seq. We end up with reads in certain regions of the genome. And so this looks like this. So you can see here a tumor sample at the top, and then the patient's adjacent bit of normal lung tissue next to it, which has peaks in different regions. And so we take that, we take 300 base pair windows is what we typically work with. And then you can see there are differentially methylated regions between conditions. So you can see there's this region here that's methylated in the tumor, but not in the normal lung. And then you can look in patients. So this is a different patient to the tumor and normal lung is coming from. But you can see there is consistency between that tumor sample from a lung tumor and then a patient, the cfDNA coming from a patient. And then it's absent in this non-cancer control sample here in cfDNA. So in terms of what we do with that data, so I started this work 4 years ago, and it's grown and grown. Initially, I had a DSL1 pipeline where it was just everything stuck together. I now have 2 separate pipelines using the NF-core tools, which are extremely useful. Like, in order to, the tools is the unsung hero, I think, to some extent of NF-core. So I have one DSL pipeline to do this demultiplexing. So because our barcodes go inside the FASTQ files, so they become the start of the FASTQ files, I end up, I have to demultiplex them to say which sample they came from originally. And because I'm impatient, the qualities talked about early, I want to parallelize that. So I actually split up the FASTQ files into chunks. And in order to run UMI tools extract, which is quite slow and single-threaded. It isn't parallelized, so I chunk it up in order to put through the same sample through these tools, and then I stick them back together and then demultiplex. Then after I've run that demultiplexing, I then have normal FASTQ files, some of which are methylation enriched, so those have the peaks. Some of them are just sort of shallow whole genome sequencing. and then I run them through a pipeline that we built in-house to analyze this kind of data. And I'm going to highlight, because I really like talking about it, the tool NGS Checkmate. I had a poster outside. So it's a tool to check that samples match when you expect them to or don't, or tells you whether they do or not. Okay, so I want to then use this for cancer of unknown primary. So these are patients with cancer of unknown primary. They have been diagnosed as having cancer, but despite a lengthy workup time, diagnostic workup, they can't figure out what original cancer type it was. So this might be because the primary site is too small to be seen through imaging in a really hard and awkward place to image, or it might have been cleared by the immune system and no longer there. Or it might just, because it's cancer and therefore has mutations, it might not be showing the typical markers, the typical things that make it look like a certain cancer. And so these patients just get standard care chemotherapy, and they don't get access to newer treatments and targeted therapies because they don't have a diagnosis. So how can we improve that diagnosis? And particularly as tissue biopsies are often very small, they're taken from the metastatic biopsy, metastatic sites, they might be quite small and necrotic, and they're used up in the diagnosis process. So we want to use cfDNA for that. So can we predict cfDNA, cancer type from cfDNA using this cfDNA methylation signal? And these methylation patterns vary across tumor types, both due to the tumorigenesis process, but also there's quite a lot of background tissue signal. So this DNA methylation is a way that your different cells with the same DNA express different proteins. So we need lots of data for this. So where do we get that from? We get that from TCGA. So TCGA, the Cancer Genome Atlas, was a massive project that sequenced about 9,000 tumor samples with methylation arrays. So that's methylation arrays. It's an older technology. It measures at fixed locations on the genome and calculates what are called beta values, measures the methylation between 0 and 1. And it's been shown, so people have taken the TCGA data and shown, oh, look, you can see tissue-specific patterns, as you might expect. So I need to convert them. I have this T7-MBD-seq profiles, and I want to do something with arrays. And so I have there's a software tool a package called QSEA that lets me go from reads to beta values. And so I do that in reverse. I go from reads to beta values to get myself a lookup table, and then I take the beta values I get from the published data and convert back to counts to make pseudo profiles for each array. And so when I do that, I take the data from TCGA, I convert it, and it's still, I get a nice UMAP sample separating by the tumor types, as you might expect. And that structure remains if I take the total array and downsample to just the most strongly different regions between each cancer type I'm interested in. And here I've got 29 different cancer types, like I've regrouped some. And so I've now got 22,300 base pair windows across the genome that I'm interested in. And I want to build a classifier. So I have these arrays that I've taken and I've converted them to be equivalent, mostly from TCGA, but a few from Gene Expression Omnibus. And I have some non-cancer control samples. That's cfDNA from from volunteers who don't have cancer. So that's the sort of you and I typical background signal. So I then do, because I'm not interested in applying this to tumor samples, I'm interested in applying this to cfDNA samples from patients, which might have 1% of their cfDNA might come from the tumor. So I do a data augmentation strategy, which is mixing, and I mix the array data into cfDNA at small amounts. So 1%, 2%, 5% to make these synthetic mixtures. And I do this lots and lots of times. I also mix non-cancer samples with other non-cancer samples, again, to increase my training population. And I end up with about 280,000 mixtures for this particular classifier. And then I subset those mixtures because like I've taken my original 2 sets of data and I've expanded them into a massive data set, which comes from all of those. So I might want to make sure I'm not overfitting. So I take subsets of that and train lots of individual classifiers. I train 100 of these sub-classifiers and then average over those. And that gives me an ensemble classifier that has 30 different tumor types, and I make predictions for a sample. All right, the classifier thinks that it's a lung cancer or a breast cancer. And the performance looks good when I apply it on sort of held out mixtures. But how does it perform on actual cfDNA samples? So I've not trained it on actual tumor samples. I've just trained it on these mixtures. And so when I apply it to actual samples, so I have 143 samples from known cancer patients of known type. So they're on the left here. So you can see the predictions that are made in the middle. I guess I should mention the classifier is called Cupid or CupID. So I have most of the predictions that I make, if I make a prediction, are correct. And then I don't make predictions on additional non-cancer controls. So I get really nice sensitivity and specificity in using cfDNA from blood samples to determine cancer type. So applying to actual patients with cancer unknown primary, I make 15 different predictions out of 41 patients. And mostly these are quite typical things you might expect in patients with cancer unknown primary. And they're clinically consistent. So these patients have a lot of diagnostic workup, and they may well get a differential diagnosis. So they're suspected to have this or this, like breast cancer or a gynecological cancer, but they're not sure which. And our classifier is generally consistent with these predictions. We will predict one of those 2. And in the patients that actually have a resolved diagnosis, so 15 of our patients get a later diagnosis. Of those, we predict the same thing in 10 of them, and 2 of them we predict something different, and 3 of them we don't make a prediction. But these take the median time to these actual diagnosis for these resolved patients is 7 months. So it's a long time of suboptimal chemotherapy and diagnostic uncertainty. So that's my actual application. But then moving back to Nextflow again, I have recently, since sort of that work, the building with that was all a set of shell scripts and R scripts. So I now have, using the NF core template and tools, a pipeline entirely custom, but using the NF core framework and structure to do the same thing. So I can build classifiers using raw IDAT files, so that's the methylation array data, if they've been deposited in the libraries, or I can use processed values of beta tables if they haven't deposited the raw data, or I can include our samples we've sequenced, and combine them together in a Nextflow way. And I can now build classifiers for different questions much easier. So to summarize, I have we have this method that can profile methylated regions across the whole genome, suitable for cfDNA. I've got these 3 different Nextflow pipelines that I've used, all based on the NF core tooling, just to emphasize how useful that is, just being able to grab the modules that you need and then build extra ones for the custom bits. And so I can convert arrays to be compatible and then build classifiers, and we can accurately classify tissue of origin for these cancer samples from plasma. And this could help in patients with cancer unknown primary. Where's my other half of my acknowledgements? Okay, there should be more on the right-hand side. Don't know where that's disappeared to. Okay, so I need to thank particularly Alicia Conway, whose PhD She's a clinician, but she did a PhD, and she did the lab samples. Alex for driving the wet lab development and optimization, and Stephen, Paddy, and Kate in particular from the bioinformatics side. And on the other half of the side, the patients who donated, like and their families who've consented, the scientific computing at CRUK, Manchester Institute, and the clinical team for collecting the samples. I think that's everybody who should be on the right-hand side. Thank you.